So in the previous class, we have seen the plasma membrane structure, its importance, right? So in today's class, we'll see various types of transport mechanisms across the plasma membrane. How exactly the transport is happening across the plasma membrane. Firstly, why is transport important, or what exactly is getting transported is various types of ions, nutrients, different molecules, water, everything gets transported in and out of the plasma membrane to and fro, that is from the cytoplasm of the cell to the extracellular matrix, from the extracellular matrix to the cytoplasm of the cell. On like both the ways the transport will take place. How does the transport take place? So there are different mechanisms for the transport of the molecules across the plasma membrane. Okay, so the types are firstly active transport. The first type is the active transport and second type is the passive transport, right? We'll first see about the active transport and then we'll move to the passive transport. What is this active transport? See, the name itself says active. Active is usage of the energy, right? Active is there is usage of the energy. So, in the active transport, the specific characteristics of the active transport is ATP is used in the transport. ATP is used in the transport, and here this transport is not along the concentration gradient, but this is against the concentration gradient. See, if there is a cell and this is all the extracellular matrix, let us imagine there are 20 molecules of sodium here. In the cell there are 20 molecules of sodium, okay, and here there are 80 molecules of sodium. Here there are 20 molecules, here there are 80 molecules of sodium. So, as a rule of the equilibrium, what should happen? So, here how many molecules have to be transferred from the extracellular matrix to the cell? What is the difference? What is the difference? How much is the difference here? 60 divided by 2 should move. Okay, always how much difference is there? Calculated. So 60 is the difference, divided by 2 is 30, right? So 30 molecules have to move here. So if 30 molecules comes inside, here it will be 50, here it will be 50. So we have achieved the equilibrium. This is along the concentration gradient, because the molecules have moved from a region of higher concentration to a region of lower concentration. So this is along the concentration gradient. Remember it, this is along the concentration gradient. Now again the same scenario, 20 Na plus and here 80 Na plus. As a rule of equilibrium, the molecules have to move from the extracellular matrix to the cytoplasm. But what is happening here is, the molecules will still move from cytoplasm to the extracellular matrix. So this is against the concentration gradient. This is against the concentration gradient. For the molecules to move along the concentration gradient, this is a natural phenomenon happening. This is a natural phenomena happening. So this does not require any extra energy here. Since it is a natural phenomena happening, it does not require any extra energy. But now this is against the natural phenomena. This is against the natural phenomena. This is against the equilibrium, right? So for this, we will have to push the movement. For pushing of the movement, the energy is been required. Energy is been required in the form of ATP. Okay, so ATP is used and the transport here is happening against the concentration gradient. Against the concentration gradient. Now, as I said, since it is happening against the concentration gradient, the movement has to be pushed, right? The movement has to be pushed. It has to be forcefully done. So, for the forceful movement, for the like, it's not happening naturally. We will have to pick, send it. Pick, send it. So for this, there are special proteins which can pick the molecules and send it across the plasma membranes. So here, in case of the active transport, along with the ATP, there is also requirement of the specialized proteins. Specialized proteins are required for the transport of the molecules across the plasma membrane. Okay. So, active transport ಅಲ್ಲಿ ATP ಜೊತೆಗೆ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಆಗಕ್ಕೆ ನಾವು ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ನ ಪುಷ್ ಮಾಡಬೇಕು ಹೌದಾ ಈ ನಾರ್ಮಲ್ ಅಲಾಂಗ್ ದಿ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಗ್ರೇಡಿಯಂಟ್ ಆಗುತ್ತಪ್ಪ ಅಂತ ಏನಾಗುತ್ತೆ it is a rule of the equilibrium that the molecules will move from a region of higher concentration to a region of lower concentration ಈಸಿಯಾಗಿ ಬಂದ್ಬಿಡುತ್ತೆ ರೈಟ್ ಈಸಿಯಾಗಿ ಬಂದ್ಬಿಡುತ್ತೆ ಅದೇ ನಾವ್ ಇವಾಗ ಫ್ರಮ್ ಅ ರೀಜನ್ ಆಫ್ ಲೋವರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಟು ಅ ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಳಿಸ್ಬೇಕಾದ್ರೆ ಎತ್ತಬೇಕು ನೀರನ್ನ right molecules agli water agli athwa ions agli nutrients agli we will have to push it right push it so for the pushing you have to apply energy illinda illige easy bidbidutte but illinda illige it has to be pushed so for the pushing you require energy okay energy jotege someone has to carry them right someone has to carry them so carrying yaar ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಅಂದ್ರೆ carrying is done by some specialized proteins like the transport proteins or channel proteins ಅಂತ ಕರಿತೀವಿ ನಾವು carrier proteins ಅಥವಾ channel proteins ಅಂತ ಕರಿತೀವಿ so we'll study about it in detail okay clear with this now there are two types of active transports okay there are two types of active transport the first one is primary active transport and the other one is the secondary active transport okay primary active transport so primary active transport alli enagate so generally whenever there is a movement of the molecule or molecules okay movement of the molecule or molecules it is just coupled with the metabolic energy that is with the ATP so normal agi just a molecule or molecules will be transported from a region of lower concentration to a region of the higher concentration that is against the concentration gradient so generally molecules coupled with metabolic energy which is in the form of the ATP. So we often study the sodium potassium pump alva sodium potassium pump alli enagatte sodium potassium pump alli so 3 sodium will be pumped out and 2 potassiums will be pumped inside right so this we call this we take as a standard example for the primary active transport so this is happening in the presence of the energy for every 3 potassiums and every 2 so for every 3 sodiums and 2 potassiums pumped in and out one ATP molecule is been utilized okay so this we call it as a sodium potassium pump which is an example for the primary active transport which is coupled with the metabolic and utilization of the metabolic energy but coming to the second type of the transport that is secondary active transport secondary active transport ಹೆಂಗಾಗತ್ತೆ ಅಂತಂದ್ರೆ let us imagine a molecule has to move uphill okay a molecule has to move uphill uphill antantandre by the endergonic reaction. What is endergonic reaction? Endergonic, exergonic. See, a cell is there. Aita? One molecule illinda illige ಬರಬೇಕಾಗಿದೆ. Okay, illinda illige ಬರಬೇಕಾಗಿದೆ. ಇಲ್ಲಿ ಬರಬೇಕಾದ್ರೆ it is using energy. Okay, it is using energy. So now if it is using energy, we call it as endergonic. Okay, endergonic, we call it as an uphill transport. We call it as an uphill transport. But ಈ secondary active transport ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, if a molecule has to undergo the uphill transport, it should be definitely coupled with one more transport of the molecule downhill. Okay, ಒಂದ್ ವೇಳೆ ಇದು uphill transport ಆಗಬೇಕು ಅಂತಂದ್ರೆ ಡೆಫಿನೆಟ್ಲಿ ಇದರ ಜೊತೆಗೆ ಇನ್ನ ಒಂದು downhill transport ಆಗಲೇಬೇಕು. ಇನ್ನ ಒಂದು ಡೌನ್ ಹಿಲ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಆಗಬೇಕು. ಸೊ ಡೌನ್ ಹಿಲ್ ಅಂದ್ರೆ ಯಾವಾಗ್ಲೂ ಹೆಂಗಿರುತ್ತೆ, ಡೌನ್ ಹಿಲ್ಸ್ ಆರ್ ಎಕ್ಸರ್ಗಾನಿಕ್. ಡೌನ್ ಹಿಲ್ಸ್ ಆರ್ ಎಕ್ಸರ್ಗಾನಿಕ್, ದೇ ರಿಲೀಸ್ ದ ಎನರ್ಜಿ. ಇಲ್ಲಿ ದೇ ಯೂಸ್ ದ ಎನರ್ಜಿ, ಬಟ್ ಎಕ್ಸರ್ಗಾನಿಕ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ದೇ ರಿಲೀಸ್ ದ ಎನರ್ಜಿ. ಇದನ್ನ ಏನಂತ ಹೇಳ್ತೀವಿ ನಾವು, ಡೌನ್ ಹಿಲ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಇದನ್ನ ನಾವು ಡೌನ್ ಹಿಲ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಈ ಡೌನ್ ಹಿಲ್, ಈ ಅಪ್ ಹಿಲ್ ಬಿಟ್ಬಿಡ್ರಿ ಒಂದ್ 2 ನಿಮಿಷ. Coming to the downhill, ಈ molecule ಏನಿರುತ್ತಲ್ಲ, let us imagine this molecule as A and this molecule as B. This B molecule, which is actually undergoing downhill, is the one which has undergone uphill reaction in the primary active transport. ಇಲ್ಲಿ ಯಾವುದು uphill ಆಗಿರುತ್ತಲ್ವಾ ಇಲ್ಲಿ ಅಥವಾ passive transport ಅಲ್ಲಿ ಯಾವುದು uphill transport ಆಗಿರುತ್ತಲ್ವಾ ಅದೇ molecule ಇಲ್ಲಿ downhill ಆಗ್ತಾ ಇರುತ್ತೆ. You got it? ಅದೇ ಮಾಲಿಕ್ಯೂಲ್ ಇಲ್ಲಿ ಡೌನ್ ಹಿಲ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಇದ್ರದ್ದು ಟೈಪ್ಸ್ ಓದ್ತೀವಲ್ವಾ ನಾವು, ಸಿಂಪೋರ್ಟ್ ಆಂಟಿಪೋರ್ಟ್ ಓದ್ತೀವಿ, ಅಲ್ಲಿ ಆಂಟಿಪೋರ್ಟ್ ಅಲ್ಲಿ ನಿಮ್ಗೆ ಇನ್ನೂ ನೀಟಾಗಿ ಅರ್ಥ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ ಸೆಕೆಂಡರಿ ಆಕ್ಟಿವ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ಅಪ್ ಹಿಲ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಅಪ್ ಹಿಲ್ ಆಗ್ಬೇಕು ಅಂದ್ರೆ ಇನ್ನೊಂದು ಮಾಲಿಕ್ಯೂಲ್ ಡೌನ್ ಹಿಲ್ ಆಗ್ಲೇಬೇಕು. ಆ ಡೌನ್ ಹಿಲ್ ಮಾಲಿಕ್ಯೂಲ್ ಯಾವ್ದಪ್ಪ ಅಂತಂದ್ರೆ ವಿಚ್ ಹ್ಯಾಸ್ ಪ್ರೀವಿಯಸ್ಲಿ ಅಂಡರ್ಗಾನ್ ಅಪ್ ಹಿಲ್. Which has previously undergone uphill. So now I repeat it once again. Secondary active transport is the one wherein one molecule is undergoing an uphill transport. For this molecule to undergo uphill transport, it should be coupled with the downhill transport of one more molecule which has previously undergone uphill transport. You got it? Which has previously undergone the uphill transport. So that is called as a secondary active transport. So in the secondary active transport, we see two mechanisms. Okay, so illi carrier proteins compulsory involved irutte, right? In all the cases we have carrier proteins involved. So the two types are, the first one is the symport and the second one is the antiport. Or in other terms we can also call them as symporters, antiporters ಅಂತನೂ ಕೂಡ ಕರೀಬಹುದು. ಇವೇನಿದು ಸಿಂಪೋರ್ಟ್ ಮತ್ತು ಆಂಟಿಪೋರ್ಟ್ ಮೆಕ್ಯಾನಿಸಮ್ಸ್, ಇವೆಲ್ಲ ಪ್ರೋಟೀನ್ಸ್ ಚಾನಲ್ಸ್ ಅಥವಾ ಕ್ಯಾರಿಯರ್ ಪ್ರೋಟೀನ್ಸ್ ಇರುತ್ತೆ ಅಂತ ಇಲ್ಲಿ ಬರ್ದಿದ್ದೀನಲ್ವಾ. ಸೊ ಸ್ಪೆಷಲೈಸ್ಡ್ ಪ್ರೋಟೀನ್ಸ್ ವಿಲ್ ಫೆಸಿಲಿಟೇಟ್ ದ ಮೂವ್ಮೆಂಟ್ ಆಫ್ ದಿ ಮಾಲಿಕ್ಯೂಲ್ಸ್, ರೈಟ್? ಸೊ ದೋಸ್ ಸ್ಪೆಷಲೈಸ್ಡ್ ಪ್ರೋಟೀನ್ಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ದಿ symporters and the antiporters. Okay, ಈ ಸಿಂಪೋರ್ಟ್ ಆಂಟಿಪೋರ್ಟ್ ಮೆಕ್ಯಾನಿಸಮ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, ಲೆಟ್ ಅಸ್ ಇಮ್ಯಾಜಿನ್ ಅ ಸೆಲ್. Okay, so cell alli now we'll first speak about the symport. Aita, symport alli enagutte, there are two molecules which are undergoing the transport. Okay, so definitely active transport alli two molecules aage agutte. So two molecules are undergoing transport. Both the molecules are transported in the same direction. Both the molecules are transported in the same direction. One example we can take, glucose and sodium. Glucose ಮತ್ತು sodium. So ಇದ್ರಲ್ಲಿ ಏನಾಗುತ್ತೆ ಸಿಂಪೋರ್ಟ್ ಅಲ್ಲಿ ಅಂತಂದ್ರೆ, ಗ್ಲೂಕೋಸ್ ಒಳಗಡೆ ಎಂಟರ್ ಆಗಬೇಕು. Okay, the glucose has to enter inside, but the glucose alone cannot enter inside. When the glucose is combined with the sodium, let us imagine ಇಲ್ಲಿ ಎಷ್ಟು sodium ಇದೆ, ಇಲ್ಲಿ 30 sodium ಇದೆ, ಇಲ್ಲಿ 70 sodium ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳೋಣ. ಆಯ್ತಾ, ಇಲ್ಲಿ 30 sodiums, ಇಲ್ಲಿ 70 sodiums. There are 30 sodiums here, there are 70 sodiums here. So as a rule, the sodium has to move from this cytosol of the cytoplasm to the extracellular matrix, right? But this is active transport. It's happening against the concentration gradient. Now what happens? The glucose has to enter, the nutrient has to enter, but it cannot enter on its own. So the nutrient combines with the sodium. Okay, this glucose combines with the sodium and both the molecules are been entering inside. So ಇಲ್ಲಿ 30 ಇಲ್ಲಿ 70 ಇದ್ರೂ ಕೂಡ ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ ಬಂತು, ಇದು 29 ಆಯ್ತು, 71 ಆಯ್ತು, ರೈಟ್? ಇದು 29 ಆಯ್ತು, ಇದು 71 ಆಯ್ತು ತಾನೆ ಒಳಗಡೆಗೆ. So this wherein both the molecules are transported in the same direction, we call it as the symport. Okay, ಇದು ಅರ್ಥ ಆಯ್ತಾ? Let me repeat it once again. So symport ಅಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ, two molecules are being transported. Okay, two molecules are being transported. Both the molecules are transported in the same direction. Both the molecules are transported in the same direction. Now let's see the beauty of this symport mechanism. Nutrients yaak transfer agate? Why the glucose is getting transferred? Because the cell needs glucose. When does the cell need glucose? If the body is already having excessive glucose, it will not need the glucose. It will not take the glucose from the extracellular matrix, right? It takes the glucose, it needs or it draws the glucose only when there is lacking, right? When it is lacking glucose, only then, when there is deficit of the nucleus, only then it takes. So here there is less amount of glucose, hence from the extracellular matrix where there is more glucose, it is taking, right? So this is fine, but for this movement we definitely require the ATP because it is a nutrient molecule. Nutrients yavattu passive transport mukantra hogak sadhya illa, ಆಯ್ತಾ? ಡೆಫಿನೆಟ್ಲಿ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ನ ಯಾರಾದ್ರೂ ಒಬ್ರು ಕ್ಯಾರಿ ಮಾಡ್ಲೇಬೇಕು, ಪ್ರೋಟೀನ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅದನ್ನ ಕ್ಯಾರಿ ಮಾಡ್ಲೇಬೇಕು, ಆಯ್ತಾ? ಸೊ ಇಲ್ಲೇನಾಗ್ತಾ ಇದೆ, ಇದಕ್ಕೆ ಡೆಫಿನೆಟ್ಲಿ ನಮಗೆ ಆಕ್ಟಿವ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಬೇಕು, ಬಟ್ ಇದು ಒಂದೇ ಬರಕ್ ಆಗಲ್ಲ. ನಾವು ಇಟ್ ಈಸ್ ಕಪಲ್ಡ್. ಓಕೆ, ಇದೇನಾಯ್ತದು ಅಪ್ ಹಿಲ್ ರಿಯಾಕ್ಷನ್ ಆಯ್ತು ನಮ್ಗೆ. ದಿಸ್ ಅಪ್ ಹಿಲ್ ಇಸ್ ಕಪಲ್ಡ್ ವಿತ್ ದ ಡೌನ್ ಹಿಲ್ ರಿಯಾಕ್ಷನ್. ಡೌನ್ ಹಿಲ್ ರಿಯಾಕ್ಷನ್ ಅಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ, ದ ಮೂವ್ ಇಲ್ಲಿಂದ ನಮಗೆ ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ ಹೋಗೋದು ನಮ್ಗೆ ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ ಬರ್ತಾ ಇದೆ. ಯು ಗಾಟ್ ಇಟ್? ಒಂದ್ ರಿಯಾಕ್ಷನ್ ಇನ್ನೊಂದು ರಿಯಾಕ್ಷನ್ ಜೊತೆಗೆ ಕಪಲ್ ಆಗ್ತಾ ಇದೆ, ಒನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಇನ್ನೊಂದು ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಜೊತೆಗೆ ಕಪಲ್ ಆಗ್ತಾ ಇದೆ. ಸೊ ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ಸಿಂಪೋರ್ಟ್ ಮೆಕ್ಯಾನಿಸಮ್. ವಿಲ್ ಕಮ್ ಟು ದ ಆಂಟಿಪೋರ್ಟ್ ಮೆಕ್ಯಾನಿಸಮ್. ಆಂಟಿಪೋರ್ಟ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಆಂಟಿಪೋರ್ಟ್ ಅಲ್ಲಿ now let us speak about this. What example we'll take? Okay, ಇಲ್ಲಿ ಸೋಡಿಯಂ ತಗೊಂಡಿದ್ದೀವಲ್ವಾ, ಈ ಸೋಡಿಯಂ ಇಲ್ಲಿ ಏನಾಗಿದೆ, ಅಪ್ ಹಿಲ್ ಆಗಿದೆ, ಹೌದಾ? ಸೋಡಿಯಂ ಇಲ್ಲಿ ಅಪ್ ಹಿಲ್ ಆಗಿದೆ. ಈಗ ಅಪ್ ಹಿಲ್ ಆಗಿರದೆ, ಸೊ ಹಿಯರ್ ವಿ ಆರ್ ನೌ ಟೇಕಿಂಗ್ ದ ಸೇಮ್ ಸೋಡಿಯಂ ಆಸ್ ಅ ಡೌನ್ ಹಿಲ್ ಮೂವ್ಮೆಂಟ್. ಸೋಡಿಯಂ ಹೆಂಗ್ ಹೋಗ್ಬೇಕು? ಸೋಡಿಯಂ ಶುಡ್ ಮೂವ್ ಔಟ್ ಆರ್ ಸೋಡಿಯಂ ಶುಡ್ ಮೂವ್ ಇನ್? The sodium should move out or the sodium should move in? Sodium heng move agate yavaglu? 3 sodiums are pumped out or pumped in? 3 sodiums are always pumped out. So avagle enaitu, the one which has come inside, the same has been pumped out. Got it? The same has been pumped out and any other molecule will be pumped in. Let us take it as a potassium only. We can take it as a potassium, right? So earlier which had undergone the uphill is now undergoing the downhill. So what does the secondary active transport mechanism means? The secondary active transport mechanism means, so here one molecule is undergoing a uphill movement. Secondary active transport andre eno, one molecule is undergoing a uphill transport which is coupled with the downhill movement of another molecule which had previously undergone uphill transport. Okay, so let me just explain the same definition with this diagram. Secondary transport is the uphill movement of one molecule. Okay, it is the uphill movement of one molecule which is combined with the downhill movement of another molecule which has previously undergone uphill movement. That's called as the secondary active transport. Okay, clear? ಏನಾದರೂ ರಿಪಿಟೇಷನ್ಸ್ ಬೇಕಾ? ಏನಾದರೂ ಡೌಟ್ಸ್ ಇದೆಯಾ ನೋಡಿ ಒಂದ್ಸತಿ. If you have not understood it at any point, you can stop me and ask. Clear? All that you have to understand here is, ಒಂದು ಅಪ್ಹಿಲ್ ಮೂವ್ಮೆಂಟ್ ಆಗಬೇಕಂದ್ರೆ ಅದರ ಜೊತೆಗೆ ಒಂದು ಡೌನ್ ಹಿಲ್ ಮೂವ್ಮೆಂಟ್ ಆಗಬೇಕು. ಆ ಡೌನ್ ಹಿಲ್ ಮೂವ್ಮೆಂಟ್ ಏನಿದೆಯಲ್ಲ, that has previously undergone uphill movement already. This is clear? Then you have understood the concept. Clear? Huh? Yes. Note it down. The next type of the transport is the passive transport. So passive transport will not include the energy. So this does not use any energy. So no ATP is being used. No ATP is used. And remember, the passive transport takes place along the concentration gradient. Like the active transport takes place against the concentration gradient, that is from a region of lower concentration to a region of higher concentration. Here the passive transport takes place along the concentration gradient, that is from a region of higher concentration to a region of the lower concentration. So it takes place along concentration gradient. Now, there are two types of passive transport. The first type of the passive transport is simple diffusion. The first type of the passive transport is the simple diffusion. In case of a simple diffusion, there are no protein carriers used. There are no protein carriers used in case of the simple diffusion, and here again no ATP used. Okay, and this is also along the concentration gradient. So simple diffusion, we can take the example of the gases. So very common example which we have studied from your previous classes are, so if you stand in a corner of a room and if you spray the perfume, in a few seconds it will take over all the room, the complete area of the room. So this is a good example for the simple diffusion, right? So simple diffusion will not involve any carrier proteins like we have seen in the active transport. There are no carrier proteins involved. There are no ATP involved. So very basic type of the movement of the molecules, we call it as a simple diffusion. Okay. Then second one is facilitated diffusion. The second one is facilitated diffusion. Of course, the facilitated diffusion also will not include any ATP. So there is no ATP used. And of course, this is also along the concentration gradient. Along the concentration gradient, But now here the carrier proteins are being used. The carrier proteins or channel proteins are used for the transport of the molecules across the plasma membrane. So carrier proteins or the channel proteins or channel proteins are used for the movement of the molecules in and out of the plasma membrane. So we'll first see the carrier proteins. Carrier proteins. One most common type of carrier protein which we see is the permease. Where have you studied the permeases? You have studied the permeases in one particular concept. Everyone have studied. It is lacoperon, right? So lacoperon alli permease ಏನು ಕೆಲಸ ಮಾಡುತ್ತೆ? There we call it as a lactose permease ಅಂತ ಕರಿತೀವಿ. But generally permease ಏನು ಕೆಲಸ ಮಾಡುತ್ತೆ ಅಂತಂದ್ರೆ, so it will induce the permeability of the molecules across the plasma membrane. So it will facilitate the permission to enter the plasma membrane. It will give the permission to the molecule to enter the plasma membrane or to cross the plasma membrane. So one example is what the permease does here is, let us imagine this is a cell. Okay, this is a cell and this is a permease molecule. Now, there is one molecule which has to enter the plasma membrane. But it cannot enter the plasma membrane on its own. Aduk swanta plasma membrane enter agake agalla a molecule ge. So ಏನು ಮಾಡುತ್ತೆ, this molecule will go and bind to the permease. It goes and binds to the permease. ಯಾವಾಗ ಪರ್ಮಿಯೇಸ್ಗೆ ಈ ಮಾಲಿಕ್ಯೂಲ್ ಬಂದು ಬೈಂಡ್ ಆಗುತ್ತೋ, this permease will undergo a conformational change. ಇದೇನಾಗುತ್ತೆ, ಒಂದು conformational change ಆಗುತ್ತೆ. I'll write it here. We'll write it in the form of the step. See, we have a permease and then we have a molecule to enter into the plasma membrane. Molecule to enter cell. Okay, to enter the cell. Now, this cannot go directly inside the cell. It cannot go into the cell on its own. So what happens? They both will combine. Permease ge bandu molecule attach agutte. Okay, when the permease and molecules bind to each other, conformational change takes place. In the permease, the molecule will remain as it is. The molecule will not take any changes. The conformational change will take place in the permease. Now, the permease will take a particular shape, attaches to the molecule and enter the cell. And it will enter the cell. Once it is entered the cell, it will release the molecule. And it goes out and comes back to its original stage. Shape. Okay, so this we call it as the carrier protein. ಇದನ್ನ ಏನಂತ ಹೇಳ್ತೀವಿ ನಾವು? carrier proteins ಅಂತ ಹೇಳ್ತೀವಿ. One very good example for the carrier protein is the glucose transport proteins ಅಂತ ಹೇಳ್ತೀವಿ ನಾವು. Okay, we call them as GLUTs, glucose transporters ಅಂತ ಹೇಳ್ತೀವಿ. So example is GLUTs or GLUT also. Glucose transporters are the best example for the carrier proteins. The second type is, okay, the second type of channels are the channel proteins. Channel proteins. Channel proteins means these are the small pores. So we have seen the structure of the plasma membrane, right? A structure of the plasma membrane alli ee tara transmembrane proteins idvu. So these transmembrane proteins or small pore-like proteins are called as the channel proteins. So these channel proteins will facilitate water, they can facilitate various ligands, they can facilitate ions, they can facilitate any molecule according to the size and the charge of the molecule. So on the basis of the size of this channel and the size of the molecule has to match. On the basis of the size and the charge on the molecule, they will facilitate the movement. So if it is facilitating the movement of the ligand, we call them as a ligand gated channel. If it is facilitating the movement of a charged molecule, we call it as a voltage gated channel. If it is facilitating the movement of an ion, we call it as an ion gated channels. If it is facilitating the movement of the water, okay, water should also be facilitated by the proteins only. Water normal diffusion alli barak agalla. Illi simple diffusion process in the water barak agalla. Water barbeku andre you remember the hydrophobic tails are in between. ಹೌದು ತಾನೆ? So ಇದು plasma membrane structure ಅಲ್ಲಿ ವಾಟರ್ ಹಿಂಗ್ ಬರ್ಬೇಕು ಅಂದ್ರೆ ಹಿಂಗ್ ವಾಟರ್ ಬರ್ಬೇಕಂದ್ರೆ ಇಲ್ಲಿ ಹೈಡ್ರೋಫೋಬಿಕ್ ಟೇಲ್ಸ್ ಟಚ್ ಆಗುತ್ತಲ್ವಾ? So water cannot enter through this hydrophobic tails. So we require specialized channels for the entry of the water also, which are being called as aquaporins. Which are called as the aquaporins. So aquaporins ಮತ್ತು ಈ ligand gated channels, voltage gated channels, ion channels, these are all the examples for the channel proteins. Okay, on the basis of their size, charge, on the basis of the size and the charge, different types of channel proteins will facilitate the movement of the molecules. Okay, and the one which is facilitating the movement of the water are called as the aquaporins. So aquaporins, these are small proteins. So these small proteins together will form one water channel or the aqua channel. Clear? Huh? Any doubts? This is about the passive transport, active transport and the passive transport. Okay? Huh? Any doubts? Note it down.